this database from microbes can be really not only for the purpose of one single company, but also potentially for a global exploration of the new novel sequences and enzymes.
Hey, Karl, how's it going?
Good, Erum. How about you?
I'm doing well. I loved our episode last week. That was fun.
Yeah, so last week, if you are just tuning in, we did an episode about biohacking, which was a lot of fun. A form of extreme biohacking done by professionals was in the news last week because a woman had her type 1 diabetes cured by the use of stem cells that I believe were injected into her pancreas. And it's the first time that this has ever happened. And given that diabetes is such a big issue, this I thought was really big news. And curing diabetes, huge news.
Oh, wow. Yeah, that is really huge. It's something we definitely have to look into. Was it instant? Was it multiple therapies over time? Will this patient now have to eat a certain way and moderate sugar? Hopefully, I mean, that is the way to live and not consume so much. But this is so great. It's great that diabetes was cured.
So this study took place in China, but the news is published in Cell, which is a prominent scientific journal. It's on the level of Science and Nature. And sure, it was a one-person, N of one study, I guess we could say. The procedure apparently only took 30 minutes. They injected 1.5 million islet cells into this woman's abdomen. She was 25 years old. 1 year later, she's still producing her own insulin. So it's lasted a year. So the stem cells took. I guess, like you say, we have to keep tracking this. But you've had a lot of other things on your mind besides biohacking. What's up?
Yeah, I've been in a lot of deep thought lately. I had mentioned to you, Karl, my son's very into space. So I've been consuming a lot of education about space and just so vast. It's so large. And just thinking about that, it's been in the news, but there's a recent circuit of news of the declassification of unidentified aerial phenomena. So then that got me into deep thinking about, wow, there could be life in outer space. And I probably have mentioned this in the podcast before because I think about that a lot. I wouldn't say I'm one of those crazy UFO nuts, but it's just that the likelihood just seems pretty clear just because not just the vastness of space, but the vastness of time. Humans have been around for only 300,000 years, but the universe has been around for 13 billion years or so. There's just so many different planets. There's planets in the Goldilocks zone. So that's ripe for life of our type to originate. So that's interesting. For me, a lot of my interest when I was a teenager thinking about what would I do, I was really interested in joining the FBI and being Scully from the X-Files. That's something that was really deep and something that I was really passionate about. And I'm starting to feel that again. I did study forensic chemistry and toxicology to pursue a career in doing autopsies on aliens, but that didn't pan out that way. But the truth is out there. And I really believe that we need to start paying a little bit more attention and listening to what's going on in the universe rather than just looking. So I think there's a whole world out there. And I probably mentioned a little bit of this when we interviewed Kate Rubins in an episode a few weeks ago. So definitely recommend listening to that one.
Yeah, I mean, it's easier these days to go down the conspiracy theory rabbit holes. I'm not going to say that all the UFO, UAP stuff is conspiracy theory.
It's conspiracy theory until it becomes conspiracy fact.
Yes. And I have definitely seen things in the sky. I've had friends who've had strange experiences while driving on country roads in Colorado. But there's a part of me that thinks, yes, there's a vast universe with life in multiple places, but I think we have alien life already on this planet. People consider octopus to be aliens in their own way. But when we look at the microbial world, we know that there's a lot of aliens there that we just don't understand or really know enough about. The number is 1 trillion microbial species. And we have very early on in the podcast spoken with Neely Ostroff, one of the co-founders of Cultivarium, which focused on making that microbial world more accessible. What's your thoughts, Erum, on the vastness of the microbial world compared to the universe?
Yeah, I mean, the vastness is very interesting in terms of microbes are everywhere. They are everywhere, cover every surface. There is no surface safe except for in hospitals and probably our homes. Unfortunately, there's not enough microbes You can't avoid them. And then there's so many different types. So you mentioned 1 trillion species, and that's just an estimate. It could be even more than that. And there's even a number to talk about the quantity of bacterial abundance, saying that there's an estimate of 5 nonillion, which is, for those that don't follow the numbers beyond a billion or a trillion, that's 5 times 10 to the 30th bacteria, making bacteria the most dominant life form by sheer number on Earth. And then they evolve, they change, they're so small, they can do things a little bit more quickly. And they do very, 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 very interesting things depending on what type of environments they live in, what they're eating, and who's around them. Because the thing about microbes is they don't live in isolation. They live in a biome. They interact with other microbes, other living things, and they sense each other. They send chemical signals to each other. There's this whole concept of quorum sensing that I've been learning about, where microbes can collaborate and make things together, like biofilms and things like that. So there's a lot, and I think we should talk about some of the weird microbes out there just to tell people a little bit about what's possible. And then I'm excited to talk about our guest today as well, who's working in the space.
All right. So what are some of the weird microbes that you flagged or that you're into?
Oh, I'm into lots of microbes, but I will talk about 3 of them that came into my sphere recently. And my sphere meaning I asked ChatGPT right before this recording about what are some weird microbes. But I've been thinking about it. Clearly, if you guys listen to other episodes, it's something that's been coming up time and time again, is that there's a whole world of microbes and the functionality is so interesting. And how do we leverage that functionality to create products or solutions for our human experience? And also these microbes, yes, they're weird, but they have superpowers. They have superpowers. They do interesting things. And there's one type of bacteria called Pele-conducting bacteria, specifically Geobacter sulforeducens, and hopefully I say that right. I mean, microbiologists are probably cringing at my pronunciation, but there we go. And Pele-conducting bacteria, as you might assume, they generate electricity and they produce electricity conductive pili, which are often called nanowires, and it allows them to transfer electrons over long distances. So this capability is used in natural environments to breathe metals like iron and uranium. So that's crazy. And scientists are studying these bacteria for potential applications in bioenergy, which is something we talked about, and to be able to create things such as microbial fuel cells. And I believe we've know some people that are working in this space.
The thing I know about these bacteria, because I had done a deep dive a few years ago on biowires or the possibility of using bacteria or biology as an alternative to electrical conductivity. And I think it said that these kind of bacteria are all over the ocean floor and sending signals to each other, which is yet another reason why we should not be mining the ocean floors. There are people out there who want to go down to the ocean floor and extract rare earth metals, but that is a bad idea. And so count me as one of those people who suggests that we do not mine the ocean floor and it should not be allowed. But anyway, keep going now that I'll get off my high horse about mining the oceans.
No, definitely agree with that. And we can certainly join a protest or anything. If you're out there thinking about mining the ocean floor, just don't do it.
So yeah, there you go.
So we talked about electricity generating bacteria, but there's also magnetism generating bacteria. There's bacteria that produce magnetic particles inside their cells, and it allows them to align with Earth's magnetic field. So that's really interesting that they're producing magnetic particles. And really, they're doing this to help orient themselves to find optimal environments. Bacteria, they don't have eyes. How do they know where to go? So they're using Earth's magnetic field to go where they need to, which I think is so fascinating.
That's nuts. Yeah. Microbial world is awesome. What else do you got?
I'll talk about the last one. And then, there's going to be millions and we're going to talk more in our episode here, but I'll talk about the last one. It's called Halomonas titanici. And I hope I said that right. Halomonas titanici. And the superpower of this microbe is titanium digestion.
What do they turn it into?
This bacteria was discovered on the wreck of the Titanic. And what it can do is break down iron and steel. So it's a part of a group of extremophiles called rust-eating bacteria. what they were doing is that they were slowly consuming the wreckage of the Titanic. And then the researchers that were looking at this bacteria, they were interested in seeing how these microbes could affect human infrastructure, because we built things with iron and steel, and specifically oil rigs and shipwrecks. But then also thinking of, okay, they're digesting iron and steel, but how can that be used in a way to do bioremediation to create healthier, say, soils or other types of remediation that is required? I thought that was really interesting that they can break down iron and steel. It's like, wow, I mean, we can't do that, but this bacteria can. So that's just one use of this bacteria, but I'm sure people with imaginations can think of how they can use Halomonas titanosi for other applications.
Yeah, speaking of imagination, I know I've said it before on this podcast, landfill remediation is going to be a $1 trillion industry, I say in the next decade, but it could be sooner, it could be later. Talking about all these bacteria makes me think not only is landfill remediation going to be a $1 trillion industry because of the stuff that we buried, but because of the microbial life that we're going to discover in these landfills. Those bacteria that are living there that have evolved around all of our garbage will potentially have superpowers that I think are going to be very useful and very valuable. But thinking about the value that we can create with these bacteria, I think is a perfect segue into our guest. Erum, do you want to talk about our guest today?
Yes, our guest, I'm so excited to have him on the podcast today, is Yuji Suzuki of bitBiome. And bitBiome is looking to discover and sequence the trillions of microbial species that are out there. And today, there's only 0.0001% of microbes that have been sequenced and examined. But bitBiome, they are going after sequencing all of them. And they have a very unique technology and platform and bioinformatics products to figure this out. They are doing an incredible job. What I also love about bitBiome is that they're based in Japan, they're international, they do work with different countries. Yuji himself is just a very interesting person, I would say, and has come from a background that's not traditional to biotech. I love to talk to people that have come into the industry from different professional backgrounds and now are coming to biotech, taking what they've learned, taking their experience and bringing it to biology and biotech. So I'm really excited for this conversation with Yuji Suzuki. Yuji Suzuki, thank you so much for coming on the Grow Everything podcast. We're so happy to have you.
Yeah, great to be here, Erum and Karl. I listen to this podcast every week, so I'm a fan of this show.
Wow, yay!
You're very kind.
I love to hear that. Oh my God. Thank you. We make it for people like you, so I'm glad you're listening. The systems are working, so this is great.
Yeah.
Yes, Yuji, we're really excited to have you on for multiple reasons, but one of the reasons that I especially like is that you do not come from a traditional biology or biotech background. Yet you're holding this leadership position at a very interesting biotech company. So you have a background in economics and international relations with prestigious posts. So Embassy of Japan, you were at McKinsey. Can you tell us how and why you made the career move from economics to biology?
Sure, happy to. The short answer is the biotech is the most exciting sector right now. But the long answer, I need to talk a bit about my personal story. I think back about 10, 15 years ago, I got seriously ill, some virus. The cause wasn't unknown, but I was almost dead and was in the hospital for about a month. And luckily, thanks to medicine, I fully recovered. But since then, I thought maybe I should know a little more about how my body works and how cells in the inner body works. And I started to learn biology from popular science. And I really then liked all the intricacies of the biology and what's going inside and around us. And I thought, oh, wow, it's the coolest thing on earth. And then I also started to learn more about biotechnology, and biotechnology can change the world in many aspects. As I get deeper into learning about biotechnology, I also learned that there's lots of development happening right now, particularly in the area of the availability of more data, because we now have more data as we couldn't have in the past. But then the harder question would be, how to utilize these newly available data and make the most of it to create a better analysis or create better models, for example, AI, and to develop the better products for a greater good. So that's why I'm really excited about biotechnology. And then here we are in bitBiome. bitBiome, the latest technology that can help companies and help the world to access to more data, particularly in the areas of microbial sequences. So I think that's now in the right position to lead the biotech world from combining my personal experience and also outside view.
You mentioned joining bitBiome. Please tell us when you joined, and then give us some background for anybody who doesn't know what bitBiome does and what it focuses on and how that applies to the bioeconomy.
Sure. I joined bitBiome 3 years ago, so it's almost 3-year working anniversary this week. To talk about bitBiome, the story actually goes back to 3.5 billion years old because microbes have been there on Earth since then. And there are right now about 1 trillion microbial species, 1 trillion. And not only they have been here around us, they have been the source of lots of innovation like antibiotics, DNA polymerase for PCR test, COVID, that saved many lives, and also bread, cheese, and wine, probably my favorite combination, and bioremediation, etc. So there are lots of things that cannot be made without microbes. But the interesting fact is that so far the identified microbes is only 10 million. That means that we have only scratched the surface of this unsung hero, 0.001%. And the remaining 99.999% is called microbial dark matter. And many companies, traditional companies and new companies, are trying to maximize the value out of this 0.001%. But we are unique in the positioning that we focus more on accessing 100% rather than 0.001%, utilizing the latest single-cell sequencing technology. So that's probably what makes us unique and unique enabler for other clients and also for the product development as a whole.
I love the fact that you have such an ambitious goal to sequence trillion microbial species, which will probably continue to evolve. We don't even know if there's a trillion. There could be more, right? But I'm so excited for bitBiome and that you have set this ambitious vision to do this and to do this with some of the most advanced technology that your company has created in-house. So it sounds like there's multiple aspects of that beyond finding these microbes and sequencing them. And can you tell us a little bit more about how you're going about this technologically? What's the process and what milestones have you achieved so far?
Exactly. bitBiome has a platform that has 3 elements. Let me go one by one. One is BitMap. It's a patented whole genome single-cell sequencing. So single-cell sequencing is different from traditional shotgun metagenomics. And if I put the analogy of solving a puzzle, single-cell sequencing is like solving one jigsaw puzzle at a time, whereas metagenomics is such as solving 100 jigsaw puzzles at one time. The result is the shotgun metagenomics itself has a great invention of 20 years old, but there are many diversities in microbes, particularly in environments such as soils, that shotgun metagenomics cannot identify the microbes accurately. But with single-cell genomics, yes, we can. So the good news is that not only can we accurately get high-quality genes using this BitMap, we can analyze the genes from microbes much, much more effectively, about more than 100 times efficiently than traditional shotgun metagenomics. That means that it would take almost 10,000 years to analyze all the microbial species in using shotgun metagenomics. But using this BitMap single-cell sequencing, it will take less than 100 years. 1 trillion species is within our reach in next generation. And secondly, we utilize this BitMap platform and accumulated sequence database with annotated and also sample source, etc., called BitGem, Gene Encyclopedia of Microbes. Now it has 2 billion genes. It's one of the most diverse and the biggest genome database in the world. And it's growing at a pace of 1 billion per year. And it only needs 2 or 3 FTEs to do that because of thanks to the continuous improvement. We started to build this BitGem about 3 years back since I joined. Once we found the potential, the speed also increased gradually. In the first phase, it took 2 years to accumulate 1 billion, but we continuously improved the protocol and workflow, and now it can grow 1 billion per year. And in the future, it may be faster. So it's a no rocket science, but having a big data is a great proposition because there are many data that nobody else can access. The third element is called BitQED, Quality Enzyme Discoverer. That's a bridge between data and product. So it's a combination of in silico bioinformatics, 3D structure analysis of the data we have, and creates proteins and enzymes in a wet lab. But I can talk about that later when we talk about more application as well.
Yeah, let's talk about the BitGem database. You say that you're able to add 1 billion sequences annually. So that would be 1 billion microbial species. What's the significance of having such a diverse and vast database? What will BitBiome be able to do with it? And are others able to access it? What would they be able to do with it?
So there are some public databases. The famous one is Magnify. It has a lot of database as well. But BitGem database has a great synergy with this public database as well. Because, for example, Magnify, the public database, most of the samples are from microbes in humans and animals, whereas about 60% of a BitGem database are from environment samples like soils and hot springs, deep seawater, and also some mine drainage, etc. So it's more diversity. And also, we can integrate the search of these two public database and our proprietary database. That means for the clients, they can find novel sequences that could be a good recipe for the food. I'm using an analogy of making a food, making a product. If clients want to make a good product, then we have to start from the good recipe. But this novel data serves as a role of good recipe.
And do you have any examples of people doing that already? Any of your partners or customers coming in, interrogating the database and finding sequences of interest that they have gone on to create things from?
Yeah. The example I can share, for example, the food company Ajinomoto is working with us, trying to identify and engineer novel enzymes. And also we have a joint collaboration with Twist Bioscience. We recently launched a transaminase kit for screening. And it dates back to SynBio Beta 2 years ago. And we made a brief presentation about this concept of BitMap and BitGem. And then afterwards, joint collaboration studies started to conduct research on how diverse our database actually is. And we find true diversity. And then we started to develop a transaminase kit enzyme used for pharmaceutical manufacturing. And we recently launched this product. It has lots of diversity than traditional transaminase. And other companies, there are many other interesting examples. And actually, some are new to biotech. What's also exciting to me is that these companies like Ajinomoto and Twist Bioscience know a lot about biotech. Then they find this novelty as attractive. But not only that, some traditional chemical companies or even semiconductor material manufacturing company reaching out to us. And in that case, we serve as a thought leader to them. And it's the application we didn't anticipate, but we found that some of the material in this hard science can be made through the power of bioprocess of microbes and enzymes. So actually, through the partnership with these clients new to biotechnology, we also find the frontiers of the how microbes can do every day.
Wow. Yeah, that's incredible. And I can imagine that you're able to have all of these microbes that have been sequenced more than most people have done. And that's really interesting because then you can identify the functionality of a specific microbe, and that might be more optimal for the application than, let's say, engineering an E. coli, which perhaps doesn't scale up to the process in which these companies require. So it's very exciting. And here at Messenger Lab, we've worked with a few enzyme design, enzyme discovery companies, and it's very challenging and difficult to develop and test enzymes. So I'm curious, how has bitBiome's technology, and particularly the bioinformatics platform, the BitQED, how has that solved the issue? We know that I'm sure you're using AI for this. So can you walk us through how that works and continue to talk about that in relation to how it impacts industries like pharma or material science? But lock it down in an analogy for us, please. Thank you.
Such a great, exciting question. The BitQED is a combination of in silico analysis. So we do bioinformatics and we do AI, and we also use 3D structure tools such as Google's AlphaFold 2, and we also do some proprietary tools to supplement that. And afterwards, we predict the points to evolve the enzymes, where to engineer. We conduct wet lab experiment using robots. And up to gram scale production is available in our wet lab right now. And next year, it will be some liters. But what's interesting is that by combining the in silico analysis in both sequence and protein structure using multiple tools, then we were able to increase the ratio to find a good starting point enzyme, good hit, which is active in E. coli host or other host, significantly. I think in the traditional culturing, it would have taken many years to find the active enzyme out of the microbes. The possibility was also maybe 1% or 2%. But using this BitQED in our latest studies, it's usually 50% to 80% to find the right hit, which is very active. Also, engineering round, they're called directed evolution, a technology developed by Nobel laureate Francis Arnold. It's a fantastic technology because it can, in the lab, evolve the microbes equivalent to tens of millions of years of evolution in the lab. So we are actually getting the future in the lab, not only just finding the path from the microbes. But anyway, it will take just 2 weeks to conduct one round right now in our lab.
Wow.
That's amazing. Amazing. Now, since you guys are working so actively in the microbial space, not just sequencing and identifying these novel microbes, you, of course, have the understanding of microbes being these incredible machines that can help address different global crises like plastic waste, carbon capture. Can you maybe talk about what the bitBiome stance is or role in advancing sustainability? How can microbes enable more sustainable solutions?
Yeah, again, we are doing multiple projects in this sustainability area. The first interesting one is plastic degrading enzymes we discovered from BitGem database. There are other companies doing this, and to a greater extent, we are also partnering with some chemical companies to conduct the discovery of engineering of these plastic degrading enzymes. The interesting thing is that usually microbes that can degrade plastics are found from where people threw away plastics, compost the soils. And about 80% of the microbes that can degrade plastics so far was found from these soils. And as our sequencing technology is particularly good at finding novel microbes from these soils, then I think not only we are discovering and engineering enzymes that can degrade plastics already, but also in the future, we can probably discover other types of microbes and enzymes that can degrade other plastics or other chemical products more effectively. That's the future dream. And the second front we are doing under the support of the government grant is to identify and enrich the variety of microbes that can eat CO2 and make products. There are some microbes already known that can digest CO2 and potentially create useful products. So it's a true dream of upcycling and killing two birds in one stone, can reduce CO2 and then not only can create some useful products for us. Every time we discuss these sustainability projects, it's in some sense quite exciting because microbes have lots of potential beyond our imagination. Of course, challenges exist in scale up and other costs, etc. But I believe that in the next 10 years can be a big game changer in this area because when I face some skepticism outside of the company, I always go back to referring the famous quote of the IBM's chairman, I think Mr. Watson in 1943. He mentioned that there will be demand for 5 computers in the world. But look now, there are lots of personal computers and mobile phones. I think the demand for this sustainability using microbes will grow beyond our expectation in the very near future.
I love that. As you know, we love to talk about how the future can play out when biotechnology takes center stage. I especially love how you brought it back to multiple benefits. It's like killing two birds with one stone. But I just heard a new phrase where they say feeding two birds with one scone. Instead, to be a little bit more fair to the birds. But my friend Marilise had said that to us. We all started cracking up on the call. And now everyone else hears it now. So that's beautiful. But the world is made of biology. The climate runs on biology. So it just makes sense that we look to biology for the climate solution. And thanks so much for that. It makes sense that the plastic eating microbes are where there's a lot of plastic. One of the things that we had talked about in the previous meeting was about finding microbes and the discovery. And you have people on your team that do that. And have you discovered microbes yourself? Have you gone out there and took your swabs and plates and gone out there and hunted for microbes?
Yeah, we have a team go to some regions to conduct collection of samples. So they enjoy some local trips and then collect samples. And not only we do that in-house, but also we have collaboration with academic institutions as well, including University of Texas. University of Texas professor is famous for plastic degrading enzymes. We have collaboration with him. And recently, also one Japanese university, University of Tokyo's team found a rock and they found microbes, 2 billion years old, literally.
Wow.
We are now conducting some analysis of this DNA to find truly novel microbes. So again, it's a very fantastic environment to find some novel enzymes. And we also actually have a government grant to set up a US lab next year to start actively collaborating more with US companies and institutions in this sample collection, because this database from microbes can be really not only for the purpose of one single company, but also potentially for a global exploration of the new novel sequences and enzymes. So that can be a good starting point for multiple industries like pharma, life science, food, nutrition, and chemical, other industries potentially as well. That's how we think we can make a greater contribution to advancing the frontier of biotechnology and SynBio.
Hi, a quick shout out to our sponsor, Messaging Lab, the force multiplier for biotech. Your biotech company is making the world a better place. You know that, we know that. But does the world? There's a big reason why some biotech companies attract investors, sign up customers, and get attention. That reason is strategic communications. At Messaging Lab, we translate complex science and economics into compelling narratives. And we have done that for the most successful biotech companies across healthcare, agriculture, personal care and beauty, materials, and the list goes on. We're here to make sure your ideas not only get heard, but resonate with your audience. So if it's time to amplify your company's voice, visit messaginglab.com to explore how we can elevate your story and grow your business.
That's amazing. One of the things that happens when you have these big databases and you're collecting these microbes, you're able to interrogate them, is that you create this platform that in turn allows AI to be trained on these models. What does that look like from, let's say, a protein design point of view, or even from a new microbe discovery point of view? How does having AI trained on this database enable those things?
Yeah, I think the power of nature, like microbes, and the power of latest technologies like de novo protein design and AI-based enzyme engineering go hand in hand, because we use AI internally and we also use enzyme engineering internally in addition to discovery. The models are always as good as the data, and there's an old proverb, cliche of garbage in, garbage out. And we conduct some joint research with an expert on protein language models and also expert on in silico protein design to see how our database can enrich the models or how we can improve these models or potentially create a disruptive models by adding the another ingredients from our database. In terms of the world of enzyme engineering, usually the experience shows that if you start from discovering a good starting point enzyme, which is highly active, high-performing enzymes, the result after engineering is also better than starting from a so-so enzyme found. So maybe if I use an analogy or baseball, my favorite sport, if you start from discovering good starting point, it's equivalent to find a good Aaron Judge or a good Shohei Ohtani in high school, rather than finding me and trying to coach me. You can find the Aaron Judge and Shohei Ohtani and coach properly. Then we can find a much better enzyme. I think that the AI model is potentially a good synergy with our database and our approach. And by having data from natural microbes and having the latest technologies such as AI, I think we can probably jointly create a much more disruptive use case for the application of these models into the real world by developing truly game-changing products.
Yeah, right. Because a lot of these models being trained on, like you mentioned, forget the number, was it 1 million microbes that are sequenced so far outside of bitBiome? So that's not a lot. The AI would have to be very creative. So being able to have diverse set of information for it to learn, and the fact that you have more microbes, it does likely create better enzymes, better proteins because it has more information to work off of.
Yeah, if you want to maximize out of this 0.001%, there's some limitation. Already, there's a lot of innovation out of that, which is great. But imagine if you can have access to 100%, then there will be maybe some novel enzyme class, or there will be novel enzyme functions that are out of reach. And one interesting example, Erum, is that when we do some analysis of how diverse our data is, we do an analysis called cluster analysis, which can illustrate how different sequences are similar each other. And we compare the sequences from BitGem and create a map of the microbe clusters, similar to the neighborhood map. But we often find some novel clusters of microbes that are only found from BitGem. So it's the different families from what we have already found using public database. So that kind of new data, new clusters can really potentially a true add value for AI analysis. And then probably more data, better models can be truly realized.
Yeah. So you mentioned clusters. I know you're talking about data. One of the things that I've been really interested in, Karl, and I've been talking about with several people, is the idea of microbial consortia, like microbiomes and how they interact. Have you done any work on microbiome dynamics and microbiome interactions and how that works?
Yeah, I think interactivity is always find harder challenge, like how microbes in soils can interact. But we recently published an academic paper where we conducted large sample survey of some human microbiomes on patient cohorts and healthy cohorts. And we found that some of the genes that can cause a resistance to antibiotics can be transferred in the form of plasmid or other DNAs. And using traditional shotgun metagenomic sequences, how drug resistance elements can be transferred to other genes couldn't be found. So that mechanism, we did a study and worked on that. But I think that the microbial diversity is a great challenge and also an exciting opportunity for us. We don't still know how the true mechanism of these microbiomes getting together and making a better benefit for us or some harms to the other earth and environment. But it's probably an exciting challenge for us in the next 5 years or 10 years because we now have an access to the, again, potentially 1 trillion species. Then how these interactions can happen is another challenge for us.
Yeah, that's amazing. Again, it's one of those things we talk a lot about, not just on the podcast, but amongst ourselves in terms of different microbiomes and how complicated they are. In fact, today we were talking about scalp microbiome and facial and skin microbiome. But thinking ahead and the future of bitBiome, what's your vision? How do you think the technology will contribute as we think about a future that maybe relies less on petrochemicals, but more on biology? How does bitBiome fit in there?
Yeah, I think that bitBiome can contribute to this creating a society less reliant on petrochemical society in multiple ways. I think there's one example I should mention of the one semiconductor manufacturing company I briefly touched upon. And they came to us. They have a commitment to reduce the CO2, and they have some agreed sustainability goal. But being a traditional chemical company, they don't know how to utilize the potential of bioprocess, green biochemistry. So we served them as a thought leader, and we conducted some search of literature and patent, etc., and found the one particular pathway that can fit for the purpose. Then the next level of challenge was to conduct the experiment to find whether it really works. And interestingly, we found a novel enzyme out of this database, which performs better than the engineered enzymes and also the publicly found enzymes. So serves the purpose. So this is still a lab scale environment, but we are probably going to go on to the larger scale. But this kind of work can show for the traditional sectors that the people think it's hard to replace by a process, then there's always a possibility to switch to the enzymatic approach or metabolic pathway-based approach. bitBiome can help not only finding the right sequences or right targets, but also start from scratch, literally. Sometimes clients come to us, hey, I want to do something. I want to do something to reduce CO2. I want to do something to be less reliant on some particular material due to the concern of supply chain. But I don't know what to do. Can you help us? Then together we create a joint team with clients and to start from scratch. Oftentimes, thankfully, we find a good potential promising candidate solution out of a database.
Wow. It's incredible. Do you have people coming to you asking for really crazy things like, hey, we want to create a bioluminescent hair dye, and we want to know if there's anything in your database? Do you have really crazy requests coming in? And have you been able to fulfill those requests?
Well, some crazy requests can come, but all crazy requests can turn out to be a truly innovative, game-changing projects. Semiconductor material case, when we start, we thought it's beyond our expectation because when we start business development, we thought that most application area of this technology is pharma, life science, and food nutrition. It's still so, but there are other areas. And hair dye, we haven't been able to have a chance to talk about skin microbes or oral microbes or other areas. Why not? And also, people's imagination can sometimes become a tool, and always the basic foundation of research starts from crazy ideas. We are happy to support any ideas, of course.
Yeah. I think a lot of people, when they're thinking of biotechnology or creating products, it's really sad. Like, oh, it just costs too much. And I think that's because they're thinking of venture capital and being able to 10x revenue. But when you have an idea, I mean, this is why designers are so interesting, and people in fashion and beauty and all these very cutting edge industries. They're just so amazing because now that we have this technology, what can we do? Right? Who's really going to push it and fund it and figure out the sky's the limit, especially when you're discovering other organisms in the deep sea that do such crazy things beyond the bioluminescence. What does that bioprocess look like? But I just think you're going to discover things, and your team is going to discover things that are just so unique. I know that you guys are focused, you guys have bitBiome, but who knows? Maybe someone on your team discovers something and they're like, you know what, this is going to be an incredible product. Bye, Yuji. I'm going to start another company. I don't know if that has happened yet. It hasn't happened.
Not right now, but recently, interesting thing is happening is that we have been encouraging people to step out of the comfort zone. For example, we established a PhD program, essentially supporting the employees to obtain PhD in universities, and we support. So after PhD, quite often companies ask these employees to remain in the company for a certain period of years, but we don't, because if he or she can get PhD using this supporting program and then start a new career, finding a company or becoming academia, then that's probably for the greater good of the society. I do hope that something wonderful discovery may lead to actual creation of a new startup or new discovery of in academic institution as well. Because at the end of the day, this technology and company is for the short term for the purpose of bitBiome's growth. But I truly believe this technology and discovery can be wide-reaching on the greater society. And we have now about 35 employees from 3 continents, the US and the UK. some reached out to us joining, and some actually migrated to Japan and then joined us. And he have been looking at us for 2 years before coming to Japan when he conducted a research on metagenomics technologies. So I think that can also help interacting with wider society of biotech and SynBio.
Amazing.
Yeah, that is amazing. You mentioned Ajinomoto. I never know if I'm saying it the right way. You mentioned Twist Biosciences. You mentioned, working with a semiconductor company. What other industries are you currently serving? And are there any industries you're not serving that you wish you did?
Yeah, right now, most of the customers are from either food and nutrition sector and chemical sector, and less from pharma and life science sector. And we also do serve academic institutions for a single-cell sequencing service. But in the future, probably thanks to the collaboration with Twist Bioscience, we are hoping to work with pharmaceutical and life science sector. Recently, some key hire was done, and I think that we can create some innovation in both the manufacturing, pharma manufacturing or process side, and potentially discovery side as well. But as I mentioned, we also do work with SynBio startups in Japan and in the US. So I think some crazy ideas or ideas further utilizing this data either to develop the model can also be interesting. The one industry sector that I hope in the future is tech-savvy companies who knows how to conduct analysis, who knows much more about the AI models. These days, the companies like Google or Apple may be looking at biotech from different angles. So different angles and different combination may also develop biotech further, because I'm from a different front, and then I'm enjoying and being excited to broaden the frontier of biotech by using this bitBiome's technology. Likewise, if I work with some traditional biotech companies and some non-traditional tech companies, then thinking about how to maximize the value of the products out of this unique database, that may be an interesting opportunity. That may be the time when East meets West and the shogun is made, like when data and model meet and some catalytic reaction can happen.
That's awesome.
Yeah.
I bet tech companies would love to work with you. I mean, the amount of computation and cloud storage required to run your programs. And oh my God, I could only imagine how much money you guys are spending on computation and cloud storage just for the databases alone.
Not to mention that we know that biology is probably in the top 3 data generating and data intensive data usage industries. So I would think the big data companies like the Metas or the Googles of the world, like you said, or the NVIDIAs would love to be able to work with bitBiome as that database grows to that size of 1 trillion species. You said in passing that you're also working with SynBio companies in Japan. What does that look like? Can you give us a preview or an idea of what some of those SynBio companies are working on?
Interesting question. There are some startups trying to create applications, and there are some biofoundries as well. One uniqueness might be there may be more focus on upcycling elements and also sustainability element, even compared to the US or Europe. Probably upcycling element using fermentation, because Japan has lots of connections with fermentation technology since ages ago, like 1,000 years ago. And traditionally, Japanese companies are world leaders in these fermentation technologies, to name a few. But many Japanese companies believe that in the traditional fermentation technology, particularly downstream technologies like scaling up, purification, they are particularly good. But in the early phase of discovery and utilizing the big data part, they still feel a bit cautious or hesitant about how it can be done. Probably because of the relative shortage of engineers or bioinformaticians or chemoinformaticians in this sector. But probably that's why companies like us, having multiple bioinformaticians and chemoinformaticians, and also have open connections with what's going on in the US or Europe, can help these Japanese SynBio companies who want to learn from the best practices of the US. And sustainability element also thanks to the government's support and government grant, because Japanese government recently launched 2 big grants in biomanufacturing SynBio. One is focusing on CO2 reduction. The other one is focusing on some upcycling. The total amount of the grant is about I think 3.5 billion USD over the next 5 to 8 years. So I think that many companies are trying to utilize this grant to create a social impact and economic gain. Again, this is how we believe that the synthetic biology can kill two birds in one stone. Or using Erum cells, maybe you have a better term now to
Feeding two
express
birds
that.
with one scone.
Yeah, feeding two birds in one scone. Yeah, I like that.
Yeah, no, that's incredible. So $3.5 billion?
Yeah.
Wow. And when did that grant release? I mean, how far along is that?
Some part is already released and we got that as well. And another application is ongoing. The commitment of Japanese government is to increase the public and private investment to 3 trillion yen, which is about 20 billion USD by 2030. So there will be lots of government and private investment ahead. That means that the investment has to increase, I think 7 times or 8 times from now. But I think some application can definitely happen in the areas of sustainability and other innovation.
Yeah. And it sounds like you know a lot of the synbio or biotech entrepreneurs in Japan. Can you tell us a little bit more about what the ecosystem looks like? I know you at BitBiome play a key role in trying to bring the ecosystem together and also bring people from other parts of the world to really foster collaboration in Japan. Tell us a little bit more about that.
Yeah, the synbio ecosystem in Japan is quite similar to the synbio ecosystem in other countries. Quite often you use an analogy of semiconductor or demarcation work. The DNA synthesis company, Twist, also has presence in Japan, and also biofoundries and companies trying to create application. I don't know if there's an example of companies trying to innovate hair dye in Japan, but there are many companies doing also fantastic things using their core technologies. In addition to that, the uniqueness is that there's one category called genome mining platform, which is us, and potentially there's one independent company. And other elements is that there are lots of university startups doing synbio research these days. So I think that's potentially similar to the US situation, but maybe more characteristics of Japanese ecosystem. Recently, there has been a growing examples of the collaboration between chemical companies and these startups, which probably can create a good virtuous cycle.
Yeah. And I was thinking more about, is there conferences or events? Are there different things? I know you guys have a SIPS and Synbio just started kicking off. How are you fostering not only for people in Japan that are already knowledgeable, but how do you bring other people, other citizens involved? How do you bring them into biotech in Japan?
Yeah. Some education from various elements is critical. I think that's why I'm a great fan of your podcast. It's a sort of the engagement to the wider outreach. I think there are multiple conferences called BioJapan or Bioengineering Conference. And these are mainly for, as you mentioned, people who are already interested in biotech and synthetic biology. To reach more wider audience from tech world or even students and grad students, I think some different effort might be necessary. And that's why we established a more casual style gathering called SIPS and Synbio, inviting the guests from abroad. And the key important element again is beer and some casual chat, because quite often the conferences tend to be very formal and also over some water. But I always admire the events in the US, such as people might casually network each other over some drinks, and the system can be probably what the Japanese ecosystem can learn from the outside of the Japan and to experience a bit more. Recently, there are some examples of startup-related gatherings where we talk about synbio. So that's probably another good opportunity to start reaching out to wider audience. Because if we go to tech conference and talk about how these microbes can actually supplement the latest technologies, then some people who know nothing about biotech may listen. That's where probably wide outreach can start.
Yeah. Thank you for mentioning that. And you're always welcome. Next time you're in New York, we'll definitely invite you to an event or have an event around you visiting and let you do a presentation about BitBiome in front of the people that we know here. And of course, we'll see you hopefully at Synbio Beta in 2025. But I'm curious, if you could envision what the future looks like in 10 years, what do you see as being the role of microbial genetics and synthetic biology in everyday life? What will be BitBiome's role in 10 years from today, in 2034?
Yeah, if I envisage 10 years from now, the first thing is that the Grow Everything podcast should be popular as ever because presumably there's a show in 25-4. That's a joke from past episodes. But the
Yes.
10 years, I like that episode. The most serious topic is that 10 years, I think that for the past 10 years has been an experiment and design DBTL cycle has been happening in the entire SynBio industry as well. So that's why we are now facing what's called the reckoning, using a more daunting word. But we also learned a great amount about how the data can be and how the better model can be and how to scale up properly. And indeed, there have been some success cases. The next 10 years, I believe that it's really to create more successful applications by leveraging what we have learned over the past 10 years, like data, model, product, scalar, application, regulatory approval. There are multiple elements. And in that, we also have some self-funded product pipelines, about 7. Some are related to pharma life science, some are related to sustainability, some related to upcycling food. But again, we are always looking at, as we develop our own products, we often know that how our technology can make a difference in accelerating the development speed, and also how we are learning from other companies or other researchers' experience over the past 10 years. And the conferences like SynBio Beta is a great example. Every year, the focus topic and changing. People come here and learn each other. So I think, again, if people are skeptical about the SynBio's future in 10 years' time, go back and look at the history of the other industries. People are quite often short-sighted and look, oh, there's no demand. But who knows? And in 5 years or 10 years, I think that there will be more great applications. And in the next 10 years, we may not even talk about SynBio, just talk about manufacturing. And the bio may become a default option rather than choose SynBio or other ways. So it's probably the question is not if, but when and in which areas.
Yes. Incredible. Love it. Shared vision. But you did forget one thing, Yuji. You forgot to talk about space. Because
yes, space microbes we are happy to analyze, and if there's anyone found in these novel sequences or any novel engineering can happen.
Yeah, in my heart of hearts, everything points to yes, there are microbes and obviously life in space. They're visiting us now. I mean, if you guys look up unidentified aerial phenomena, it's insane how much news is already out there and what the US government is starting to share with the public. Again, I know I always go crazy at the end of these interviews, but it's something that if you want to look for unique space microbes, I mean, there's going to be an opportunity there soon.
Absolutely.
Yeah, it's awesome, isn't it? We know a lot about innovation or potential microbes. Then new discovery comes and we then learn how less we know. So it's a continuous learning process, isn't it?
Yeah,
That makes
it really
this
is.
sector exciting. Yeah.
Yes. And then you start getting curious about the new information and you just continue going down these thought experiments. Then you can talk about it on a podcast. So here we are.
Yeah.
Yeah, when wisdom of microbes and the latest technology of mankind meets, great things can happen.
Absolutely. Well said. Well said. So, Yuji, I just want to say thank you so much for coming on to the Grow Everything podcast. We will certainly have you on again. I know we'll be talking offline. It's just fascinating what you guys are doing. 100% of microbes being sequenced, being used and understood. Ambitious vision. And thank you guys, you and bitBiome, for carrying on this vision.
Yeah, likewise. Karl, Erum, thank you very much. It's great to be here and enjoy the show.
Awesome.
Thank you, Yuji.
So, Karl, what did you think of that episode?
Yuji's an incredible person, just period. And bitBiome is an incredible company. As we were getting ready to start the interview, we talked a little bit about his experience at McKinsey, and I revealed that couple of my favorite strategy books were written by a McKinsey executive from Japan. These are books that were written in the 80s or early 90s. But hearing about bitBiome and what bitBiome is doing is incredible. They are an incredible resource. And just thinking about what you're going to be able to do when you can access the genomes or just being able to access this database of all these microbes, I think is going to be incredible. We talked about it at the top of the hour. We talked about interviewing Neely of Cultivarium. And Cultivarium is a focused research organization. So all the knowledge that they gain gets made public. And I think Yuji made it clear that many of the things that they're doing are also going to be publicly available. So I think what bitBiome is doing is an incredible service. And I think being able to access what they have is going to be incredibly powerful. And I think you joked around about it, Erum. Have you had people who have left to start a company based around a bacteria? I'm sure that's going to happen.
Oh, yeah. Yeah. Sounds like Yuji's encouraging that with his team by funding their PhDs. Like, wow, what a great culture to really enable your team to think bigger, think more deeply, pursue their passions while they're also helping grow the company. And I think that says a lot about leadership as well as culture in companies. And I just love when companies do that. We've been going down these rabbit holes about microbiomes and being able to understand the different species that work together. And this whole concept of single cell sequencing is not concept. This whole technology of single cell sequencing is amazing. It's really changing the game because then you can figure out what microbes have the functionality, taking a look at them and how they interact with other microbes. I think that's something that could really help understand functionality, but also bring those microbes into commercial applications versus, let's say, engineering a microbe to do something that's not naturally accustomed to versus finding a microbe that can just naturally do it. And what does that mean for industrial applications? Maybe instead of forcing an E. coli to do something it's not naturally designed to do, creates all these inefficiencies, but having a microbe that naturally produces the thing, the compound, it can scale up more easily with less cost, right? This is something that we like to think about from the business standpoint because we work with all these companies in the space that are looking to make bioproducts, bio-based ingredients, biomaterials, therapeutics, and how do they do so at a cost that makes sense so they can then sell it at a cost that consumers are able to pay. So I just love that. And it's so fundamental. Think of, wow, there's trillions of species of microbes out there. They're doing all sorts of amazing things. And being able to formulate a question saying, hey, look, I want to create something that does this. What type of microbes can do this? Just think of it like Gen AI and being like, okay, I'm looking for a microbe. I'm doing my do. That's me typing to ask a question of some Gen AI. I don't want to have any allegiance to any of them, but to say, hey, look, I'm looking for a microbe that can make my hair glow. What are the top 5 microbes that I can cultivate? Give me the protocol to cultivate the microbes, where I could find them, what media is required to feed them, and how can I make it into an application? And then AI just spits out everything that I need to take the steps to make my dream a reality. I didn't need to hire all these different people to figure that out. I just had to query bitBiome's database or Cultivarium's database, and being able to have that is where the future is going. I think it's wonderful. We live in a wonderful time right now.
Yeah, I mean, I think, I'm going to bring it back to what we talked about at the top of the hour. The answer is out there. And
Yeah.
Cultivarium and bitBiome are making these answers accessible to us all. And I think, like you said, it's super exciting. Something else that is super exciting is we're getting close to our 100th episode. Our next episode is going to be our 99th, which I think we're going to spend some time reflecting on what we've done or learned in these first 100 episodes. And then we have a very special guest for the 100th, which we're not going to tease out yet. But I think that with that, we should just remind you listeners to become a Patreon, join our community. It's like having a cup of coffee with your best friends in biotech. If you don't know anything about what we do or what we talk about or who we've interviewed, you should definitely do that and let us know. We have an 800 number where you can leave us messages, questions, and that's the Grow Everything hotline. And you should get in touch with us. Did I miss anything, Erum?
No, you checked up all the boxes.
All right, that's the pod.